Imagine you take, just an everyday allergy pill to stop a runny nose. And then 30 minutes later, you are practically unconscious on the sofa.
Right, a very common experience.
Yeah, but then consider a totally different scenario. You take a medication specifically designed to stop terrifying auditory hallucinations. And a few weeks later, your hands will not stop trembling when you just try to hold a cup of tea.
Two very different clinical pictures.
Exactly. Why do these seemingly disconnected things happen? Well, today we're looking at the invisible master tuners of the brain that cause all of this. We are taking a deep dive into the absolute structural spine of psychiatric training.
We are indeed. We're getting into Kaplan and Sadok's comprehensive textbook of psychiatry. Specifically, we're looking at chapter 1.7, which is all about biogenic amine neurotransmitters.
And we should establish right away, for you listening, we are not here to merely recite a textbook or give you like a numbered list of receptors to memorize.
Oh, absolutely not. That wouldn't be very helpful. We are here to build robust mental models. We really need to clarify these complex mechanisms and strengthen your clinical reasoning.
Right. We want to look at how psychiatrists actually think about the brain, moving from the mechanism to the manifestation and ultimately, to the clinical meaning.
Because ultimately, psychiatry is the study of how brain, mind, and experience interact. And crucially, how that process can go wrong.
Spot on. Okay, let's unpack this with an analogy. I want you to imagine the human brain as a massive, just unfathomably complex symphony orchestra.
Okay, I like this.
So the amino acid neurotransmitters, things like glutamate and GABA, they are the string section and the percussion. They play the main melody. They set the basic tone and the primary rhythm.
Right, they're the fundamental on and off switches of the brain's electrical signaling.
Exactly. But the biogenic amines, the ones we are talking about today, they are the conductor. They are the acoustics of the hall itself. They are essentially the master mixing board at the back of the auditorium.
That is the crucial distinction here. Biogenic amines do not just transmit raw data. They modulate it. They shape the nuance of the signal.
They tune things.
Yes, they tune our mood, our motivation, our attention, our arousal, and really our very capacity to assign meaning to the world around us.
Which is huge.
It is. They adjust the brain's overall state. So, perhaps turning up the brass section when we need hyper-alertness in a crisis, or muting the percussion to allow us to fall into deep sleep.
So you really cannot begin to understand a psychiatric disorder or its pharmacological treatment without understanding these master tuners.
You really can't.
And by understanding these modulators, specifically dopamine, noradrenaline, serotonin, acetylcholine, and histamine, you will suddenly understand why psychiatric medications do what they do.
You'll see exactly why a drug might successfully cure a delusion, but then cause massive unexpected weight gain as a side effect.
So let's establish the core conceptual framework here. Who are the main characters in this biochemical cast?
Well, we are looking at a highly specific family of molecules.
Oh.
First up, we have the catecholamines.
Okay.
And that is a subgroup that includes dopamine, noradrenaline, which you might also know as norepinephrine, and adrenaline, or epinephrine.
Got it.
Then, standing slightly apart structurally, but functionally related, we have serotonin, histamine, and acetylcholine. These are our biogenic amines.
Now, I'm struggling with a fundamental concept here. It's something the text calls the topographical paradox.
Oh, yes.
Because if these chemicals control absolutely everything from my working memory to my basic drive to get out of bed in the morning, they must be everywhere, right? Like they must just coat the entire cerebral cortex in a thick layer.
Well, that is the logical assumption, certainly, but it is anatomically completely incorrect.
Wait, really?
Yes. The cell bodies of these neurons, the actual biological factories where the neurotransmitters are synthesized and where the cell's nucleus resides, they're highly restricted.
Where are they then?
They are not scattered throughout the cortex at all. They are clustered into incredibly tiny, highly concentrated nuclei. And these are primarily hidden away in the evolutionary older parts of the brain. So the midbrain, the brainstem, the basal forebrain, and the hypothalamus.
So they are essentially trapped in these tiny clusters at the very base of the brain. How do they control the whole symphony from way down in the orchestra pit? Aren't their signals getting severely bottlenecked?
Well, the secret lies in their arborization.
They're branching.
Exactly. Their axons, the long wire-like projections carrying their electrical signals, they project massively and extensively across the entire central nervous system.
Like a tree.
Very much like the root system of an ancient sprawling tree.
Yeah.
reaching all the way up into the highest levels of the cortex, diving deep into the limbic system, wrapping around the cerebellum, and even descending down the spinal cord.
That is incredible.
This incredibly wide disbursement is what permits a tiny cluster of neurons to modulate activity in diverse, distant brain circuits all at once in a highly coordinated fashion.
The text actually has a statistic that completely broke my understanding of cellular biology.
Oh, the synapse count.
Yes. It estimates that a single human neurostriatal dopamine neuron has over 1 million synapses.
It is staggering.
One cell literally reaching out and touching a million other cells.
It is an absolute architectural marvel. Just to put that in perspective for you, that is at least 2 orders of magnitude greater than what we see in standard cortical neurons.
Wow.
But, and this is crucial, that massive, sweeping architecture comes with a profound biological cost.
An energetic cost, presumably. I mean, maintaining a million connections can't be cheap for a single cell.
It requires astronomical amounts of energy, and furthermore, these specific axons are unmyelinated.
Meaning they don't have that insulation.
Right. They lack the fatty insulating myelin sheath that helps electrical signals travel efficiently in other parts of the nervous system.
Oh, that sounds incredibly demanding.
It is. Maintaining over 1 million synaptic connections on a single unmyelinated neuron places the cell under a phenomenal metabolic burden. It requires constant, massive mitochondrial activity and oxygen consumption just to keep the membrane potential stable.
And clinically, I imagine that makes them the weakest link in the chain.
Highly susceptible, yes.
Yeah.
Susceptible to biochemical, environmental, and pharmacological insults.
So if anything goes wrong, they are the first to suffer.
Precisely. If the brain experiences oxidative stress or is exposed to a neurotoxin, or honestly even just undergoes the normal wear and tear of aging, these specific neurons are uniquely vulnerable. They simply burn out.
And that explains clinical diseases.
It does. This fundamental architectural vulnerability helps explain the pathophysiology of Parkinson's disease, for instance.
Where you lose dopamine.
Exactly. In Parkinson's, we see a profound, progressive, and highly specific loss of these neurostriatal dopamine neurons, while the neighboring cells remain entirely healthy.
Let's put the listener in that reality for a second. Think about what that means for you. A tiny cluster of cells at the base of your brain, working so hard that they are practically on the brink of metabolic collapse, is literally reaching out and touching millions of other cells to decide if you feel motivated today, or if your hand trembles when you reach for a door handle.
It's a very fragile balance.
So to understand how we try to fix that balance when it breaks, we really need to look at the life cycle of these neurotransmitters. Let's look at the assembly line, the synthesis.
Okay, let's start there.
The text points out that dopamine, noradrenaline, and adrenaline all share a common biosynthetic pathway. They are built from the exact same raw materials. How does this start?
Well, it actually begins with what is on your plate.
Diet.
Yes. Blood-borne tyrosine, which is an amino acid derived from dietary proteins, circulates in your bloodstream. However, it cannot just slip easily into the brain.
Because of the blood-brain barrier.
Exactly. It has to cross the blood-brain barrier via a highly specific gateway. It's called the large neutral amino acid, or LNAA, transport system.
Okay, so it gets a ride on the transporter.
Right. And once it passes that barrier and enters the brain's extracellular fluid, it is actively taken up into the catecholamine neuron itself.
And once it's inside the factory floor, the manufacturing begins.
Yes. In the cytosol of the neuron, an enzyme called tyrosine hydroxylase goes to work. It essentially converts tyrosine into L-DOPA. And this is vital. This is the crucial, rate-limiting step of the entire catecholamine assembly line.
Meaning it paces the whole operation. It's the bottleneck.
That's right.
So if you need more dopamine quickly, you can't just dump more tyrosine into the system. You have to wait for tyrosine hydroxylase to do its job.
Precisely. And because it is the slowest step, it is the most tightly regulated by the body, and also the most susceptible to pharmacological manipulation.
Okay, so we have L-DOPA now.
Yes. Once you finally have L-DOPA, another enzyme steps in. It's called aromatic amino acid decarboxylase. We commonly abbreviate that as AADC.
Much easier to say.
Much easier. So AADC rapidly converts L-DOPA into dopamine. And if we are sitting inside a dopaminergic neuron, well, the process just stops right there. Dopamine is the final finished product.
But the assembly line doesn't stop there for the other catecholamines, does it? If dopamine is the finished product for one neuron, what happens in a noradrenaline neuron? Does it just keep building on top of the dopamine?
It does. It adds to it. Noradrenergic neurons express an extra enzyme called dopamine beta hydroxylase, or DBH.
Okay.
And here's a really fascinating quirk of cellular geography. DDH isn't just floating freely in the cellular fluid. It is actually anchored inside the storage vesicle.
Oh, the little storage bubble.
Yes. So the newly synthesized dopamine has to be actively transported into a protective vesicle, and only inside that little lipid bubble does DBH convert it into noradrenaline.
Okay, so the dopamine gets packed into a vesicle, DBH converts it to noradrenaline inside that protective bubble.
Whoop.
But what about adrenaline? Because that's the next step, right? For adrenaline, does the cell just add another enzyme inside the vesicle?
You know, it seems like that would be the logical, efficient design, but no.
Oh.
In adrenaline-producing neurons, there is one final enzyme. It's called phenylethylamine N-methyltransferase, or PNMT.
Quite a mouthful.
It is. The quirk here is that PNMT is located back out in the cytoplasm. It requires a specific methyl donor called SAMely, which is only available outside the vesicle.
Wait.
Yeah.
So the noradrenaline has to be pumped back out of the safety of the vesicle into the cytoplasm, get methylated into adrenaline by PNMT, and then be pumped back into a vesicle for final storage.
That is exactly what happens.
That sounds terribly inefficient and risky. I mean, the brain is literally pumping precious neurotransmitters back and forth across membranes just to access different tools.
It really does sound inefficient.
It's like a car factory where you build the chassis, lock it in a shipping container, realize you forgot the steering wheel, take it back out of the container, add the wheel, and lock it back up again.
That's a brilliant analogy. It is a profound demonstration of evolutionary patchwork, really. The identity of the neuron, whether it is dopaminergic, noradrenergic, or adrenergic, is dictated entirely by which of these specific enzymes it genetically expresses.
That structural reality explains so much about our clinical treatments. Like, the textbook brings up Parkinson's disease treatment. We established that Parkinson's is driven by a lack of dopamine in the striatum.
Right.
So the most obvious question any medical student asks is, well, why don't we just give patients a dopamine pill to replace what's missing?
Because dopamine itself is structurally incapable of crossing the blood-brain barrier.
Ah.
If you swallow a dopamine pill, it will simply circulate in your peripheral bloodstream. It will cause systemic side effects like massive fluctuations in your blood pressure, but it will never actually reach the brain where it is desperately needed.
So it's useless for Parkinson's.
Essentially, yes.
Yeah.
So instead, we administer a precursor. We give L-DOPA, usually prescribed as levodopa.
Well, because L-DOPA is earlier in the assembly line.
Exactly. L-DOPA is a neutral amino acid, so it can use that LNA transport system to easily cross the blood-brain barrier.
The same one tyrosine uses.
Spot on. And more importantly, giving L-DOPA entirely bypasses the slow, rate-limiting tyrosine hydroxylase bottleneck.
Oh, that makes sense.
By supplying exogenous L-DOPA, we ensure that once it gets inside the brain, the abundant AADC enzyme can rapidly convert it into a massive supply of dopamine.
But there's a catch here, right? We mentioned AADC is abundant. It isn't just in the brain, it's everywhere in the body.
That is the primary pharmacological hurdle we face. If you administer levodopa on its own, the AADC enzyme present in peripheral tissues like the lining of your gut and your liver will rapidly convert almost all of it into dopamine before it ever has a chance to reach the brain.
So it gets converted too early.
Right. And this causes a massive, dangerous spike in circulating peripheral dopamine.
And peripheral dopamine causes severe nausea. The text points out that it forcefully activates dopamine receptors in the area postrema.
Yes, the area postrema.
Which is the brain's vomiting center. And crucially, it sits structurally outside the protection of the blood-brain barrier, so it can monitor the blood for toxins.
Exactly. So to prevent this catastrophic nausea and ensure the drug actually reaches the central nervous system, levodopa is never given alone.
What do we mix it with?
It is routinely administered with a peripheral AADC inhibitor, usually something like carbidopa.
Okay.
The clinical brilliance of carbidopa is that it strongly blocks AADC, but its molecular structure prevents it from crossing the blood-brain barrier.
Oh, it's an escort.
That's a good way to look at it.
The carbidopa acts as a bodyguard in the bloodstream, fighting off the AADC enzyme so the L-DOPA isn't converted prematurely, preventing the systemic nausea.
Mm-hmm.
But then, once they hit the blood-brain barrier, the carbidopa gets turned away at the door, and the L-DOPA slips inside to be converted into dopamine right where the patient actually needs it.
It represents one of the truly great triumphs of rational drug design in neurology. And it's based entirely on knowing where these specific enzymes live.
That is incredibly elegant clinical reasoning. Now, what about the other side of the factory? We've covered the catecholamines. What about serotonin? Is it a similar assembly line?
It relies on a similar conceptual framework, yes. but it uses a different starting material.
Not tyrosine.
No, serotonin synthesis does not start with tyrosine. It begins with a different dietary amino acid called tryptophan.
Okay.
Tryptophan also crosses the blood-brain barrier using that exact same LNAA transport system we just talked about.
Oh, the same gateway.
Exactly. And this creates a fascinating physiological dynamic because tryptophan constantly has to compete with other large neutral amino acids, including tyrosine, to secure a seat on the transporter into the brain.
Wait, does that mean what I eat actually changes the competition at the blood-brain barrier? Like, if I eat a massive steak, am I flooding the gates with competing amino acids and inadvertently lowering my brain's serotonin production?
The relationship between diet and central neurochemistry is definitely complex, but essentially, yes. Dietary protein and carbohydrate ratios can specifically influence brain tryptophan levels by altering plasma amino acid patterns.
That is wild.
For example, carbohydrate consumption triggers insulin release. Insulin pulls competing amino acids into muscle tissue, leaving tryptophan a much clearer path to the brain.
Which explains so much.
Right. This is actually a proposed mechanism for carbohydrate craving in atypical depression. Your body is trying to clear the path for tryptophan.
It was so clever.
So once inside the serotonin neuron, an enzyme called tryptophan hydroxylase converts tryptophan to 5-hydroxytryptophan, or 5-HTP.
I've seen that as a supplement.
Yes, you have.
Ugh.
And then that same versatile AADC enzyme we saw in the dopamine pathway steps in to decarboxylate 5-HTP into serotonin, which is technically known as 5-hydroxytryptamine, or 5-HT.
And the textbook mentions that in one highly specific region, the pineal gland, the assembly line, actually keeps going to make melatonin.
It does. In the pineal gland, serotonin is not the final product. It is further metabolized by acetylation and O-methylation to form melatonin. Melatonin is then secreted into the bloodstream to regulate our circadian rhythms in direct response to the environmental light and dark cycle processed by our retinas.
Okay, so the neuron has successfully manufactured these biogenic amines. They are sitting there in the cellular fluid, the cytoplasm. But the text makes it very clear they aren't safe just floating around in there, are they?
No, not at all. The cytoplasm is a highly hostile environment for a free-floating neurotransmitter.
Why is that?
It contains an aggressive enzyme called monoamine oxidase, or MAO.
Ah, MAO.
And MAO is physically anchored to the outer membrane of the cell's mitochondria. MAO is essentially a cellular shredder. Its sole biological purpose is to oxidize and destroy free-floating biogenic amines to prevent toxic accumulation.
So how does the neuron protect its precious inventory from its own shredding machines?
It sequesters them.
Hides them away.
Right. The cell utilizes a specialized transporter protein called the vesicular monoamine transporter, specifically VMAT2 in the central nervous system.
Okay, VMAT2.
VMAT2 sits on the membrane of those storage vesicles we mentioned earlier. It acts as an active pump. It literally burns energy to pull the transmitters out of the dangerous cytoplasm and pack them safely inside the protective lipid vesicles. So they're ready to be released into the synapse when an electrical signal actually arrives.
And interfering with that specific pump has massive historical pharmacological implications. The text brings up an old drug called reserpine. We really should talk about how observing this one drug practically built the foundation of modern psychopharmacology.
Reserpine is a fascinating piece of medical history. It is an alkaloid derived from Rauwolfia serpentina, which is the Indian snakeroot plant.
Okay.
In the mid-20th century, Western medicine began using it extensively to treat severe hypertension.
High blood pressure.
Exactly. It was highly effective at lowering blood pressure. But physicians soon noticed a devastating side effect in a significant subset of their patients. They developed profound, vegetative clinical depression.
And mechanistically, looking at the cell diagram, we now know exactly why, right? Reserpine was irreversibly blocking VMAT2.
It was. By blocking VMAT2, the vesicles were rendered entirely incapable of storing monoAs.
Oh, no.
So the newly synthesized dopamine, noradrenaline, and serotonin were left stranded in the cytoplasm. And what's waiting for them there?
The MAO shredders.
Exactly. They were systematically shredded by MAO. The neurons were left completely depleted of their neurotransmitters.
And that caused a depression.
Observing this phenomenon that a drug-depleting biogenic amines directly cause severe depression practically birthed the monoamine hypothesis of mood disorders, which posits that depression is fundamentally linked to a deficiency in these specific master tuners.
That is just incredible history. Conversely, amphetamines do the exact opposite, right? They don't deplete the stores, they sort of weaponize them.
Amphetamine-like drugs disrupt this carefully managed sequestration completely. They force their way into the neuron, they collapse the vesicular pH gradient, and they mobilize the neurotransmitters out of the storage vesicles back into the cytoplasm.
Just dumping them out.
Right. Then, they force the primary plasma membrane reuptake transporters to run in reverse.
Oh, wow. So it pumps them out of the cell entirely.
Yes, actively pumping massive, unregulated amounts of dopamine, noradrenaline, and serotonin out of the cell and into the synaptic cleft.
Which brings us nicely to the synapse itself, the microscopic gap between communicating neurons. So the neurotransmitter is released into this gap, and it has to bind to a receptor on the target cell to pass the message. Let's talk about the nature of these receivers.
Well, the receptors for biogenic amines broadly divide into two fundamental architectures. We have G-protein coupled receptors, or GPCRs, and ligand-gated ion channels.
Okay.
It is absolutely vital for any clinician to remember that the vast majority of the receptors we cover in this chapter are GPCRs.
Right, the GPCRs are the true modulators. They don't just act like a simple door opening to let electricity in. They send a messenger down to the boiler room to change how the whole building operates.
That is an excellent way to conceptualize it. When a neurotransmitter binds to a GPCR, it doesn't open a pore. Instead, it changes the shape of the receptor.
A conformational change.
Exactly, which activates a G protein inside the cell. This initiates a slow, complex, modulatory second messenger cascade.
Like what?
Well, it might increase or decrease the production of cyclic AMP, or perhaps activate phospholipase C, which in turn alters gene expression and protein phosphorylation.
So it's changing the cell structurally.
It is. A GPCR alters the fundamental metabolic state and future excitability of the target cell. It's a slow, profound shift. However, there are two critical exceptions among the biogenic amines.
Let me guess. The fast switches.
Yes, the serotonin 5-HT3 receptor and the nicotinic acetylcholine receptors.
So these are not GPCRs.
No, these are ligand-gated ion channels. When the respective transmitter binds, a central pore snaps open immediately, allowing specific ions to flood into the cell and generate a rapid, millisecond-scale electrical signal. They are built for speed, not slow modulation.
Now, I have to ask about a terminology distinction that always trips up students. Autoreceptors versus heteroreceptors. We hear these terms constantly in psychopharmacology. So an autoreceptor is like a thermostat for the neuron itself. It just shuts off the furnace when the room is warm enough?
The thermostat analogy is perfect. An autoreceptor is located on the presynaptic terminal, or sometimes on the cell body, of the very neuron that released the transmitter.
Okay, so it's checking its own output.
Crucially, yes.
Yeah.
It responds specifically to that neuron's own neurotransmitter. It functions as a local negative feedback inhibition mechanism.
Can you give an example?
Sure. If a dopaminergic neuron is firing rapidly and there's a surplus of dopamine flooding the synapse, some of that dopamine binds to the presynaptic dopamine autoreceptor. This triggers a signal inside the neuron, telling it to hyperpolarize, stop firing, and halt further dopamine release. It prevents the whole system from burning out.
And a heteroreceptor, how is that different?
A heteroreceptor is a receptor for a particular neurotransmitter, but it is located on the terminal of a neuron that produces a completely different transmitter.
Oh, I see.
For example, you might find a specific serotonin receptor located on the terminal of a dopamine-producing neuron.
Okay.
When serotonin binds to that heteroreceptor, its activation can either increase or decrease the release of dopamine. This is the actual physiological mechanism by which the different sections of the brain's orchestra communicate with and dynamically modulate each other.
That makes total sense. Okay, so the signal has been sent, the receptors are activated. How do we turn the system off? Because if these chemicals just sat in the synapse forever, we'd be in a constant state of overstimulation. How do we clean up the mess?
The brain employs dual systems of synaptic cleanup. We have reuptake pumps and enzymatic degradation.
Let's start with reuptake.
For dopamine, noradrenaline, and serotonin, the primary, most efficient method of signal termination is reuptake. The presynaptic neuron is equipped with high-affinity plasma membrane transporters, so DAT for dopamine, NET for noradrenaline, and SERT for serotonin.
And they just pull it back in.
Yes, they act like highly specific cellular vacuum cleaners, rapidly recapturing the released transmitter from the synaptic cleft and pulling it back into the presynaptic terminal.
To be recycled.
Exactly. Once inside, it can be repackaged by VMAT2 for future use or destroyed by MAO if it's no longer needed.
And this vacuum mechanism is exactly where some of the most famous, widely prescribed psychiatric drugs in history do their work.
Precisely. Selective serotonin reuptake inhibitors, or SSRIs like fluoxetine or sertraline, physically bind to and block SERT.
They block the vacuum.
Right. Serotonin noradrenaline reuptake inhibitors, SNRIs like venlafaxine, block both SERT and NET.
And illegal drugs do this too, don't they?
They do. Drugs of abuse exploit this exactly. Cocaine aggressively blocks D8, intensely prolonging dopamine's actions in the brain's reward pathways. By jamming the vacuum cleaner, these compounds leave the neurotransmitter in the synapse for extended periods, massively amplifying and prolonging its modulatory signal.
And if the vacuum doesn't catch it, the enzymes are waiting. We already met MAO, but there's another major player mentioned in the text, COMT.
Ah, yes. Catechol-methyltransferase, or COMT. While MAO handles intracellular breakdown, COMT is another crucial enzyme, primarily responsible for degrading catecholamines in the extracellular space.
So outside the cell.
Yes. In clinical practice, COMT inhibitors like entacapone are frequently prescribed alongside levodopa in the management of Parkinson's disease.
Why is that?
Because by inhibiting COMT, you slow the peripheral and central breakdown of levodopa. This prolongs its half-life, smooths out its clinical effects, and actually reduces the total dose requirements for the patient.
It is incredibly satisfying when the mechanistic puzzle pieces snap into place like that. Okay, let's take this dense molecular foundation and do a deep dive into the clinical manifestations of each specific transmitter.
Let's do it.
Let's start with the undisputed superstar of neuroscience media, dopamine. You know, pop culture constantly tells us dopamine is simply the pleasure chemical. You get a like on your social media post, you get a hit of dopamine, but the textbook paints a vastly more complex, nuanced picture.
It does. Reducing dopamine to merely pleasure is a gross oversimplification. Dopamine is fundamentally about salience, action, and learning. It tells the brain what is important and drives the motivation to pursue it.
So it's about importance, not just feeling good.
Exactly. And to understand dopamine clinically, you must separate it into its distinct anatomical pathways. There are three major pathways we really must discuss: the nigrostriatal, the metalimbic and mesocortical, and the tuberoinfundibular pathways.
Let's start with the one we touched on earlier, the one with the million synapses, the nigrostriatal pathway.
Right. This pathway originates in the substantia nigra, located in the midbrain, and projects heavily to the dorsal striatum, which comprises the caudate nucleus and putamen.
And what does it do?
This specific circuit is deeply involved in the unconscious regulation of motor control, the selection and smooth initiation of physical actions, and the slow, iterative establishment of motor skills and habits.
So when this specific pathway undergoes degenerative cell death, we see the classic hypokinesia, the extreme rigidity, and the resting tremor that defines Parkinson's disease.
Yes, the loss of that tonic dopamine modulation leaves the striatum entirely out of balance, unable to properly initiate movement.
Right.
But equally important for the psychiatric trainee is what happens when we block this pathway artificially.
With drugs.
Yes. All antipsychotic drugs, to varying degrees, rely on blocking dopamine D2 receptors to treat psychosis. If a clinician prescribes an antipsychotic that blocks D2 receptors in the dorsal striatum too aggressively, they induce iatrogenic Parkinsonian-like side effects in their patient. So, stiffness, slowed movements, and tremors.
And what if that aggressive blockade continues for years?
Well, the system attempts to adapt. The striatum upregulates its D2 receptors, becoming hypersensitive.
Oh, that sounds bad.
It is. This long-term exposure can lead to tardive dyskinesia, a notoriously difficult-to-treat condition that manifests as repetitive, involuntary, hyperkinetic movements, most commonly grimacing, lip-smacking, or chorioathetoid movements of the tongue and mouth.
Right, because the brain is desperately trying to amplify a signal that the drug is blocking. Now, what about the mesolimbic and mesocortical pathways? Because these seem much closer to the core of psychiatric pathology.
They are. These pathways originate adjacent to the substantia nigra in a region called the ventral tegmental area, or VTA.
Okay.
The mesolimbic projections target subcortical limbic areas, most notably the nucleus accumbens. Now, this is often labeled the classic reward pathway. But in modern neuroscience, it is far better understood as mediating goal-oriented behaviors, assigning incentive salience to environmental cues, and calculating reward prediction error.
Let's pause and define reward prediction error, because the text heavily emphasizes this concept, and it is crucial for understanding addiction and learning. Give me a concrete example.
It is the fundamental algorithm by which the brain learns to navigate the world. Imagine you are playing a slot machine. If you expect a small reward and you receive exactly what you expected, the baseline firing of your dopamine neurons remains entirely steady. There is no new information to learn.
But what if I hit the jackpot unexpectedly?
If you expect a small reward but receive a massive, unexpected jackpot, you experience a positive prediction error.
Okay.
Your VTA dopamine neurons fire in a rapid, massive burst, flooding the nucleus accumbens with dopamine. Subjectively, this feels intensely rewarding, but biologically, it is a learning signal. It stamps in the memory, screaming at the brain, whatever you just did to get that, remember it and do it again.
And if I pull the lever expecting a payout and the machine just eats my coin?
A negative prediction error. You expected a reward and got nothing. In response, the tonic firing of your dopamine neurons actually pauses.
It dips.
Yes. The dopamine levels briefly drop. This temporary deficit signals the brain to abandon that specific behavior. It is a dynamic, continuous learning system.
And what about the mesocortical pathway?
Those are projections from the VTA extending up into cortical regions, particularly the prefrontal cortex. This noradrenergic, dopaminergic interaction is absolutely essential for executive function.
Like planning.
Exactly. Sustaining working memory, maintaining task goals over time, complex decision-making, and behavioral flexibility.
So if we look at the most severe psychiatric illnesses, how does this relate to schizophrenia? Because for decades, we've heard about the dopamine hypothesis. The idea was simply that schizophrenia was caused by too much dopamine.
Right. The oldest iterations of the dopamine hypothesis did posit a generalized hyperactivity. But through neuroimaging and clinical observation, we now understand it is vastly more nuanced.
It's not just too much everywhere.
No, it is a problem of topographical imbalance. The acute psychotic symptoms, the terrifying auditory hallucinations and paranoid delusions are strongly associated with increased dysregulated dopamine release in the associative and dorsal striatum.
So the brain is assigning intense, aberrant salience to ordinary stimuli, leading to paranoia.
Precise.
But what about the negative symptoms, the severe apathy, the emotional blunting, the cognitive decline?
Those symptoms are actually associated with reductions in dopamine release in the prefrontal cortex and the ventral striatum.
Wow.
This is why prescribing a massive dose of a first-generation antipsychotic, which blindly blocks dopamine receptors everywhere, might cure the hallucinations but actually worsen the apathy and cognitive dulling. The patient is no longer hallucinating, but they are left entirely devoid of motivation.
It's a cruel paradox. Too much dopamine in the striatum causing chaos, not enough in the cortex to plan a path out of it. Okay, the third dopamine pathway, the tuberoinfundibular pathway. This one isn't about thought or movement at all. It's about hormones, right?
Yes. These specific dopamine neurons are located in the arcuate nucleus of the hypothalamus and project a very short distance to the median eminence.
What do they do there?
Their entire job is to release dopamine directly into the local portal blood circulation, feeding the anterior pituitary gland. Here, dopamine acts as a profound hormonal inhibitor. It tonically binds to lactotroph cells and suppresses the release of prolactin.
Which perfectly explains a very common, highly distressing side effect of our antipsychotic medications. If you give a patient a drug that aggressively blocks D2 receptors to stop their psychosis, you inadvertently block that crucial inhibition in the pituitary.
Correct. Without that tonic dopamine break, prolactin levels rise dramatically, resulting in a condition called hyperprolactinemia.
And what does that look like clinically?
Clinically, this manifests as galactorrhea, which is inappropriate breast milk production in men or women, also significant gynecomastia, severe menstrual disturbances, and profound sexual dysfunction.
From the drugs?
Yes. Drugs with a very tight, unyielding affinity for the D2 receptor, like haloperidol or risperidone, are notorious for inducing this, whereas drugs that bind less avidly or act as partial agonists, like aripiprazole, are largely sparing of this pathway.
Here's where the neurobiology gets really interesting for me. The text notes that dopamine neurons don't just fire at one speed, they operate in two completely different physiological modes: a slow, steady, pacemaker tonic mode, and a rapid burst phasic mode. Why? Why does the brain need two gears for the exact same chemical?
It is a brilliant mechanism for coding different types of information. The different rates of firing translate to entirely different meanings for the receiving brain regions.
Give me an example.
At the fastest scale, burst firing or phasic activity elicits a massive, transient, high-concentration spike in synaptic dopamine. This phasic burst is exactly what signals the reward prediction error we just discussed.
The jackpot.
Right. It is the aha moment. It is the sharp alert, something highly unexpected and salient just occurred.
And the slow, steady mode. What is the point of just dripping dopamine constantly?
That is the tonic mode. It is a slow, rhythmic, highly regulated pacemaker activity that maintains a steady baseline concentration of extracellular dopamine in the brain.
So just humming along.
Yes. This conic level isn't about acute learning. It enables sustained, long-term motivation, ongoing complex cognitive processes, and the smooth, continuous execution of motor control.
Ah, so this is what fails in Parkinson's.
Exactly. It is this foundational tonic-enabling function that degrades and fails in Parkinson's disease. And recent neurophysiological data suggest these modes actually exist on a functional continuum, where post-burst elevations gradually sustain tonic functions, seamlessly linking the acute motivation of a reward with the sustained cognitive effort required to secure it.
So, to summarize the dopamine system, it is a sprawling, multi-pathway network coding for absolutely everything from physical movement to motivation, reward prediction, and hormonal regulation, all firing in dynamic rhythms to give salience and meaning to our experiences.
That's a great summary.
Let's shift our focus to its close chemical cousin, noradrenaline or norepinephrine.
Right. While dopamine is the engine of salience and reward seeking, noradrenaline is primarily the architecture of arousal, vigilance, and the body's global physiological response to stress.
The textbook pinpoints an incredibly tiny cluster of neurons for noradrenaline. The locus coeruleus, or LC, situated in the pons of the brainstem.
Yes, the locus coeruleus is astonishingly small, yet from this tiny nucleus, noradrenergic fibers project to virtually all regions of the brain. The entire cerebral forebrain, the thalamus, the cerebellum, and descending down the spinal cord.
That's a massive reach.
It is. An unusual and highly significant feature of this network is that a single noradrenergic neuron typically branches to innervate several anatomically distinct brain regions simultaneously.
Talk about a global tuning system. So if the locus coeruleus fires, it doesn't just wake up the visual cortex, it wakes up the entire central nervous system in unison.
Exactly. It coordinates a unified state change. And similarly to dopamine, LC neurons exhibit state-dependent activity modes.
The two gears again?
Right. Increased tonic, steady background activity in the LC is associated with periods of wakefulness, behavioral flexibility, and broad environmental scanning.
And the burst mode.
Conversely, short periods of intense burst, or phasic firing, are specifically linked to highly arousing, unexpected stimuli, shifting the brain from broad scanning to focusing attention sharply on a specific, immediate threat or vital task.
So if my LC is firing constantly in a high tonic mode, I'm highly awake, flexible, but perhaps easily distracted by every little noise. But if it bursts, I drop everything and hyper-focus on the tiger jumping out of the bushes.
That is a very apt translation of the survival biology, yes.
Yeah.
Clinically, psychiatrists and physicians interact with noradrenaline through adrenergic receptors, which historically were divided into alpha and beta classes.
The text breaks down the clinical pharmacology beautifully here. Let's look at the alpha-1 receptors first, because we usually think of these in the emergency room, not the psychiatric clinic.
True. The predominant peripheral effect of alpha-1 receptor agonists is the potent constriction of arterial smooth muscle, driving up blood pressure.
Oh, like Sudafed.
Exactly. This is why alpha-1 agonists are the active ingredients in over-the-counter nasal decongestants. They constrict the swollen blood vessels in the nasal mucosa to clear the airway.
But in psychiatry, we use alpha-1 antagonists, right? Drugs like prazosin.
Yes. Prazosin effectively blocks alpha-1 receptors. While it was primarily developed as an antihypertensive medication, it has proven uniquely useful in treating specific autonomic symptoms of post-traumatic stress disorder.
Specifically nightmares, I believe.
Particularly trauma-related nightmares. By blocking central alpha-1 receptors in the brain, prazosin blunts the excessive, terrifying noradrenergic reactivity that is triggered by trauma reminders during sleep, allowing the patient to rest without waking in a state of sheer panic.
That's a profound use of pharmacology. Then there are the alpha-2 receptors. These are fascinating because functionally they act as the brakes on the system.
Yes, alpha-2 receptors predominantly function as G-coupled autoreceptors.
The thermostats again?
Exactly. When noradrenaline binds to an alpha-2 autoreceptor on the presynaptic neuron, it initiates a signaling cascade that strongly inhibits the further release of noradrenaline. Therefore, when we administer an alpha-2 agonist, like clonidine or guanfacine, we are actually tricking the system into turning itself down, reducing central sympathetic tone.
They have a major therapeutic role in disorders involving prefrontal cortex dysfunction, don't they?
They do indeed.
Like ADHD and Tourette syndrome.
Correct. The prefrontal cortex relies heavily on an optimal, highly specific level of noradrenergic tone to maintain working memory and executive function. Too little, and you are distracted. Too much, and you are panicked.
So where does guanfacine come in?
Guanfacine, which is highly selective for the alpha-2A subtype, improves working memory by binding to postsynaptic alpha-2A receptors in the prefrontal cortex. This strengthens the signal-to-noise ratio in cognitive networks, allowing a patient with ADHD to hold on to a thought without losing it to a distraction.
And clonidine.
Clonidine, a broader alpha-2 agonist, is heavily employed to manage the agonizing acute phase of opioid withdrawal.
Oh, right.
Opioid withdrawal is characterized by a massive, uncontrolled, hyperactive noradrenergic storm originating right there in the locus coeruleus. Clonidine steps in, hits the alpha-2 autoreceptors, and forces the locus coeruleus to calm down, alleviating the sweating, tachycardia, and severe agitation.
And finally, the beta receptors.
Beta-adrenergic antagonists, universally known as beta blockers like propranolol, are mainstays in cardiology for managing hypertension and arrhythmias. But psychiatrically, they are highly effective non-sedating tools for reducing the peripheral physical signs of increased sympathetic activity.
Like stage fright.
Exactly. The severe hand tremor, the profuse sweating, and the rapid pounding heartbeat associated with performance anxiety, social phobia, or stage fright. They physically block the body's escalating panic response, which in turn helps calm the psychological panic, breaking the feedback loop.
Okay, we are moving on to the third major modulator, serotonin. Now, when the general public hears serotonin, they instantly think the brain chemical for happiness. But the text drops an absolute bomb right at the start of this section. Over 80% of the body's serotonin is located in our gut.
It is a remarkable, often overlooked biological reality. The vast majority of serotonin is produced by enterochromaffin cells lining the gastrointestinal tract.
Doing what?
There, its primary job is to modulate gut motility, secrete fluids, and manage digestive functions. In the actual human brain, only roughly 1 in a million neurons produces serotonin.
1 in a million.
Yes. Yet this evolutionary ancient molecule projects so widely that it influences virtually all aspects of central nervous system function.
Yeah, wait, hold on. Let me interrupt with a clinical question here. If 80% of my serotonin receptors are in my gut, and I take a standard SSRI antidepressant to boost serotonin in my brain, am I accidentally flooding my digestive tract with serotonin? Is that why gastrointestinal distress and severe nausea are such massive immediate side effects when you start an antidepressant?
That is exactly what is happening. The SSRI does not know to only target the brain. It blocks the serotonin transporter systemically.
Oh, man.
The sudden surge of serotonin in the gut hyperstimulates the enteric nervous system, leading directly to the nausea, cramping, and diarrhea that cause so many patients to prematurely abandon their medication in the first week. It is a perfect example of how understanding the whole body distribution of these transmitters explains the clinical reality of our drugs.
Okay, but let's look at that crucial 1 in a million fraction located in the brain. Where are they hiding?
They are clustered exclusively in the midline Raphe nuclei of the brainstem.
The Raphe nuclei?
Right. The caudal or lower Raphe nuclei project downwards into the spinal cord, heavily regulating pain perception and autonomic processes. The rostral or upper nuclei, specifically the dorsal and median rive, project extensively upwards into the entire forebrain, modulating mood, anxiety, aggression, feeding behaviors, and the sleep-wake cycle.
The anatomical distinction between the dorsal and median raphe projections is fascinating to me. It's not just a uniform blob of serotonin spreading upwards, they have completely different architectures.
The topography is highly differentiated. The serotonergic fibers originating from the dorsal raphe are very fine, with small fusiform varicosities.
And the median.
Conversely, the fibers from the median raphe are much thicker, punctuated with large beaded varicosities. While their projection fields overlap significantly in the cortex, they innervate completely different subcortical areas.
And crucially, they have completely different vulnerabilities to toxins. The text explicitly mentions MDMA, commonly known as ecstasy.
Yes, MDMA is an amphetamine analog that induces massive, immediate vesicular release of serotonin, resulting in its euphoric and empathogenic effects.
But there's a danger.
A significant one. Neuroanatomical studies demonstrate that MDMA causes long-lasting, potentially permanent neurotoxic damage, specifically to the fine, vulnerable axons of the dorsal raphe, while entirely sparing the thick, beaded axons of the median raphe. This specific neurotoxic vulnerability highlights the deep structural and functional heterogeneity within the serotonin system itself.
Now, to really understand serotonin pharmacology, we have to look at its receptors. And the text notes there are 14 different identified serotonin receptor subtypes.
A huge family.
We obviously can't cover them all without turning this into a 5-hour lecture, but let's deep dive into the heaviest hitters. Let's group them by their clinical story. Let's start with the primary thermostat, the 5-HT1A receptor.
The 5-HT1A receptor is widely distributed throughout the brain and frequently acts as a G-coupled autoreceptor located on the soma and dendrites of the serotonin neurons themselves, strongly inhibiting cellular firing.
So it turns serotonin down.
Right. Drugs that act as partial agonists at the 5-HT1A receptor, such as buspirone, are clinically useful non-sedating anxiolytics. Furthermore, activating 5-HT1A receptors can robustly augment the efficacy of traditional antidepressants.
How does that work?
The theory is that they initially desensitize these inhibitory autoreceptors, eventually allowing for greatly enhanced forward serotonergic signaling over time.
Okay, now for the 5-HT2 family. This is arguably the most important family in modern psychiatry. Let's talk about 5-HT2A. I like to think of this one as the psychedelic receptor.
It's the primary mediator of psychedelic states. The profound alterations in sensory perception, visual processing, and the deep sense of meaning induced by classic hallucinogens like LSD, psilocybin, and mescaline are directly mediated by full or partial agonism at the 5-HT2A receptor, which is highly concentrated on apical dendrites of cortical pyramidal neurons.
But psychiatrists don't just study it for psychedelics, they actively target it every day with antipsychotics.
Yes, the entire class of second-generation or atypical antipsychotics is defined mechanistically by their relatively higher potency as antagonists of the 5-HT2A receptor compared to their affinity for the dopamine D2 receptor.
Let's unpack that. Why does blocking a psychedelic receptor help treat the paranoia and hallucinations of schizophrenia?
Because the monoamine systems do not exist in isolation. Blocking 5-HT2A receptors on downstream neurons profoundly modulates dopamine release in specific pathways.
Ah, crosstalk.
Exactly. It is thought to improve the negative and cognitive symptoms of schizophrenia and dramatically reduce the risk of the extrapyramidal motor side effects, like the Parkinsonism we discussed, that inevitably occur when you just blindly block D2 receptors everywhere.
Are there drugs that just hit 5-HT2A?
We actually also have newer drugs like pimavanserin, which is a highly selective inverse agonist, specifically at 5-HT2A. It is approved to treat the debilitating hallucinations and delusions in Parkinson's disease because it calms the psychosis without blocking dopamine at all, thus sparing their already compromised motor function.
That brings us to a darker side of the 5-HT2 family, 5-HT2C. The text points to this one as the primary metabolic brake, and blocking it causes a hugely frustrating tug-of-war in clinical practice.
It is a critical receptor for understanding side effects. 5-HT2C receptors are intricately involved in appetite suppression and metabolic regulation.
So it tells you to stop eating.
Yes. When we prescribe highly effective atypical antipsychotics, most notably clozapine and olanzapine, they unfortunately have a very high antagonistic affinity for the 5-HT2C receptor.
So they block the brake.
Blocking this receptor essentially removes the biological brake on appetite. Patients experience insatiable hunger, driving rapid, significant weight gain, insulin resistance, and eventually metabolic syndrome. Furthermore, 5-HT2C receptors in the VTA tightly regulate dopamine release in the nucleus accumbens, meaning this receptor sits right at the complex intersection of mood, reward, and compulsive feeding.
And finally, the oddball of the serotonin family, 5-HT3, the vomiting switch.
The 5-HT3 receptor is entirely unique. It is the only serotonin receptor that is a ligand-gated ion channel rather than a slow GPCR.
Fast switch.
It mediates incredibly fast excitatory transmission. It's densely located in the medulla of the brainstem, particularly in that area poststramal we keep returning to.
Right, the brain's toxin detector.
Exactly. 5-HT3 antagonists, most famously ondansetron or Zofran, are highly effective antiemetics used globally to treat the severe nausea associated with cancer, chemotherapy, and surgery. By physically blocking the fast 5-HT3 ion channel, you stop the emetic signal instantly.
And you avoid dopamine.
Crucially, yes, because it targets serotonin and not dopamine, you prevent the nausea without running any risk of the Parkinsonian side effects you get from using older dopamine D2 antagonists like metoclopramide for nausea.
Okay, we've thoroughly covered the big three: dopamine, noradrenaline, and serotonin. Let's move to acetylcholine. This is the memory and motor balance transmitter. Where is the acetylcholine factory located?
Central cholinergic neurons originate primarily from the basal forebrain complex, most notably the nucleus basalis of Meinert, which projects heavily to the cerebral cortex to support memory and learning. There is also the mesopontine complex in the brainstem, which innervates the thalamus to regulate arousal. Additionally, there is a small but incredibly important population of cholinergic interneurons entirely contained within the striatum, where they counterbalance dopamine.
The synthesis is relatively simple, just one step combining choline and an acetyl group. But what's highly unique is how acetylcholine is cleaned up from the synapse. It refuses to use a reuptake vacuum.
That is a critical structural distinction. Termination of cholinergic transmission is primarily dependent on immediate aggressive enzymatic breakdown right there in the synaptic cleft. It's done by the enzyme acetylcholinesterase, or ACE.
So no vacuum at all.
No. The enzyme acts like molecular scissors, instantly ripping the acetylcholine molecule into inactive choline and acetate. Almost half the choline is then pumped back up into the presynaptic cell to be recycled.
And this rapid breakdown is the exact target for our primary Alzheimer's medications.
Yes. In Alzheimer's disease, the pathology involves a significant, devastating degeneration of the cholinergic neurons in the nucleus pacellus of Meinert, leading to profound cortical cholinergic deficit, which drives the memory loss.
So we want more acetylcholine.
Centrally acting reversible ACE inhibitors like donepezil, rivastigmine, or galantamine physically block the breakdown of acetylcholine. This prolongs its presence in the synapse, attempting to compensate for the dying cells and modestly improving cognitive symptoms.
The acetylcholine receptors are split into two completely different families, muscarinic and nicotinic. Let's start with muscarinic. These are the slow GPCRs.
Yes, there are 5 subtypes, M1 through M5. The prototypical muscarinic antagonists are naturally occurring alkaloids like atropine and scopolamine, derived from the highly toxic, deadly nightshade plant, Atropa belladonna.
The text has a great, morbid historical note here. Belladonna literally means beautiful woman in Italian. During the Renaissance, women in Italy would squeeze the toxic berry juice directly into their eyes to block muscarinic receptors and chronically dilate their pupils, which was considered highly attractive.
Indeed, blocking parasympathetic cholinergic tone aggressively dilates the pupil. But when administered systemically, muscarinic antagonists cause a massive cascade of parasympathetic blockade effects.
The anticholinergic toxidrome.
Exactly. Severe dry mouth, painful constipation, blurred vision, urinary retention, and, crucially for psychiatrists, profound central cognitive impairment, confusion, or florid delirium. Medical students learn the mnemonic: blind as a bat, mad as a hatter, red as a beet, hot as a hair, dry as a bone.
Which is incredibly relevant for us because so many of our psychiatric drugs are accidental, clumsy muscarinic antagonists.
Yes, many older tricyclic antidepressants, first-generation antipsychotics, and even over-the-counter first-generation antihistamines possess highly significant antimuscarinic properties.
So that's the dry mouth.
This is exactly why psychiatric patients constantly complain of dry mouth and severe constipation, and why geriatric patients who already have age-related cholinergic decline are highly susceptible to medication-induced delirium. However, we also use muscarinic antagonists intentionally.
Oh, we do?
Drugs like benztropine are prescribed to treat the severe motor symptoms of Parkinsonism induced by antipsychotics, aiming to restore the delicate balance between dopamine and acetylcholine in the striatum.
Then there are the nicotinic receptors. These are the fast ion channels.
They are ligand-gated ion channels constructed of 5 distinct subunits arranged around the central pore. They're widely distributed throughout the brain. The powerfully reinforcing addictive effects of inhaled nicotine depend entirely on its interaction with these receptors in the brain's reward system, specifically promoting the burst release of dopamine from the VTA.
And we use rationally designed drugs to target this for smoking cessation, like varenicline, commonly known as Chantix.
Varenicline is a brilliant piece of pharmacology. It is a partial agonist with high selectivity at the alpha-4-beta-2 nicotinic receptor subtype. It partially stimulates the receptor just enough to relieve the agonizing withdrawal cravings, but because it binds so tightly, it blocks full nicotine from a cigarette from binding if the patient relapses and smokes.
It just sits in the seat.
Exactly. There's also immense ongoing research interest in the alpha-7 nicotinic receptor as a therapeutic target for improving the severe neurocognitive dysfunction seen in schizophrenia and Alzheimer's.
Okay, the final major player of the classical biogenic amines is histamine. If I'm honest, I mostly think of histamine when I have hay fever and my eyes are watering. But it is a vital central neurotransmitter too.
It is deeply important. Histamine-containing neuronal cell bodies are restricted exclusively to the tuberomammillary complex of the posterior hypothalamus. Like the other amines, they send highly diffuse unmyelinated projections throughout the entire brain. Their primary physiological role is the rigorous regulation of sleep, central arousal, and appetite.
And this finally explains the Benadryl effect I mentioned at the very beginning. When I take diphenhydramine for a pollen allergy, it crosses the blood-brain barrier, blindly blocks the H1 receptors in my hypothalamus, completely shuts down that critical arousal signal, and that's why I pass out on the couch.
Exactly. Central blockade of H1 receptors results in profound, undeniable sedation. This is a very common and often highly unwanted side effect of many psychotropic medications, particularly antipsychotics like clozapine and quetiapine, and antidepressants like doxepin.
So sedating.
In fact, doxepin's heavy sedative property is so potent that it is sometimes used therapeutically in extremely low doses, specifically to treat primary insomnia.
But what about non-drowsy allergy meds?
Newer allergy medications like loratadine are specifically engineered structurally so they cannot cross the blood-brain barrier, allowing them to treat peripheral allergies while entirely avoiding the central sedation.
Are there other histamine receptors we target psychiatrically?
Centrally, the H3 receptor is exceptionally interesting. It functions as a presynaptic autoreceptor, inhibiting further histamine release. Recently, a highly selective inverse agonist at the H3 receptor, a drug called pitolisant, has been developed and approved.
And what does that do?
By forcefully blocking the H3 autoreceptor's inhibitory signal, it tricks the neuron into releasing massive amounts of wake-promoting histamine. It is proving highly effective for treating the excessive diurnal sleepiness and cataplexy seen in narcolepsy.
Before we move to the final integration, we absolutely have to touch on the trace amines. The text includes a short section on this, but it truly sounds like the next great frontier of psychiatry.
Trace amines, such as phenylethylamine, tyramine, and tryptamine, are structurally very similar to our classic biogenic amines, but they exist in the brain in extraordinarily low concentrations. For decades, we simply didn't know much about their function until the recent discovery of trace amine-associated receptors, or TARs. The TAR1 receptor is highly expressed in the VTA and the dorsal raphe.
The textbook mentions studies with genetically modified mice where the TAR1 receptor is entirely knocked out. What happens to them?
They become hyperactive. Activation of the TAR1 receptor seems to tonically inhibit dopaminergic and serotonergic neurons. If you knock it out, those neurons fire far more rapidly. TAR1 seems to act as a crucial intracellular negative regulator of neurotransmission, possibly by forming functional complexes with dopamine or serotonin autoreceptors.
And how are we using this clinically?
This is the absolute frontier right now. TAR1 agonists are currently undergoing late-stage clinical trials as a completely novel treatment paradigm for schizophrenia.
Wow.
They modulate and calm dopamine firing indirectly from inside the cell membrane without ever blocking the D2 receptors directly. If successful, this means we could treat psychosis with a drug that carries no risk of Parkinsonism, no risk of tardive dyskinesia, and no abuse potential. It represents a fundamental shift in psychopharmacology.
So we have covered an immense amount of molecular biology. What does this all mean for the practicing clinician or the medical student trying to make sense of a patient sitting in front of them? Let's synthesize this into clinical reasoning.
The ultimate clinical takeaway is that expert psychiatrists do not look at isolated neurochemical deficits. They do not think, depression equals low serotonin, therefore I will add serotonin.
Right, it's not a plumbing problem.
No. They look at profound shifts in perception, motivation, arousal, and meaning. These biogenic amines are vast, intricately interconnected tuning networks. Dysfunction here does not break one specific thought. It causes global shifts in how the brain interprets the entire world.
The art of psychopharmacology, then, is an exercise in complex receptor binding profiles. It's about finding the right key that hits multiple locks at once.
Exactly. Atypical antipsychotics like clozapine or aripiprazole are often affectionately referred to in the literature as dirty drugs, but in a highly beneficial way.
Dirty in a good way.
Yes. They don't just hit one single target. They carefully, dynamically balance D2 blockade, 5-HT2A antagonism, perhaps partial 5-HT1A agonism,
Right.
all while trying to minimize dangerous off-target histamine and muscarinic effects. The psychiatrist is attempting to restore a functional global balance across a massively dysregulated symphony.
And it is not just isolated switches acting alone. You mentioned TAR1 might form complexes. The text explicitly mentions the concept of receptor heteromers. What are those and why do they matter?
Historically, neuroscientists assumed that GPCRs functioned as single, entirely isolated units floating in the cell membrane. We now have undeniable evidence that different receptors can physically bind together to form complex structures called heteromers.
Give me an example.
For instance, a D2 dopamine receptor might physically interlock with a 5-HT2A serotonin receptor, or even a cannabinoid receptor, to form a completely unique hybrid signaling complex with entirely novel functional properties.
Wait, the switches are literally talking to each other before the signal even enters the cell.
Yes. The integration of the signal happens at the membrane level. It vastly multiplies the complexity of the brain's computational power.
That is incredible. Okay, let's tag team a rapid-fire summary of our key takeaways from this massive foundational chapter. Number 1, biogenic amines act as master modulators, not just simple on-off signals.
2, they regulate global, overarching brain states, mood, motivation, attention, and arousal.
3, dopamine is the engine mediating salience, reward prediction, and motor initiation.
4, serotonin is the great stabilizer, regulating mood balance, sensory perception, and emotional inhibition.
5, noradrenaline governs alertness, vigilance, and the body's acute adaptation to stress.
6, the architectural scale is staggeringly massive. Tiny, highly vulnerable nuclei in the brainstem and midbrain project millions of unmyelinated synapses across the entire central nervous system.
7, dysfunction in these systems causes broad, devastating shifts in worldview and behavior, not isolated neurological deficits.
And 8, modern psychiatric treatments work by targeting multiple receptors simultaneously, attempting to restore functional, delicate balance across these complex, interacting tuning systems.
Brilliant. Now let's end with a final provocative thought.
If we now know that receptors physically link up to form these unique heteromers, like a dopamine receptor, literally binding to a serotonin receptor to create a totally novel hybrid signaling unit, what if the future of psychiatry isn't about flooding the whole brain with serotonin or blindly blocking all D2 dopamine receptors?
What would it be?
What if the future is designing highly specific molecules that target only the specific heteromer driving a single pathological symptom, leaving the rest of the brain's beautiful symphony entirely untouched?
That would change absolutely everything. No more blunt instruments. True precision tuning. Okay, before we let you go, let's test your clinical reasoning. We have 5 clinical vignettes to reinforce the mental models we just built. Let's put you, the listener, in the clinic. I'll describe the scenario and ask the question, give you a moment to pause your player to think, and then we'll unpack the clinical reasoning behind the answer.
Sounds good.
Here's scenario 1. You are treating a patient with Parkinson's disease. You know that to improve their symptoms, you need to increase dopamine in the brain. However, you cannot simply prescribe a dopamine pill because it won't cross the blood-brain barrier. Instead, you prescribe levodopa. Which specific enzyme, which acts as the crucial rate-limiting bottleneck in the normal synthesis of dopamine, are you intentionally bypassing by giving levodopa? Take a second to think.
The answer is tyrosine hydroxylase. Under normal conditions, tyrosine hydroxylase converts tyrosine to L-DOPA. Because it is the absolute slowest step in the entire catecholamine synthetic pathway, it acts as the primary bottleneck. By supplying L-DOPA directly, you bypass this strict control point, allowing the abundant downstream enzyme, AADC, to rapidly synthesize the dopamine your patient desperately needs.
Scenario 2. A patient with chronic schizophrenia has been on a high dose of a typical first-generation antipsychotic for 10 years. During a routine clinical review, you notice they have developed severe, repetitive, involuntary movements of their mouth, tongue, and jaw. Which specific dopaminergic pathway's long-term blockade is primarily responsible for causing this distressful side effect? Take a second to think.
The answer is the nigrostriatal pathway. This vital pathway projects from the substantia nigra to the dorsal striatum to control smooth movement. Long-term aggressive D2 blockade here forces the receptors to upregulate and become hypersensitive, completely disrupting the balance of motor control. This leads to the hyperkinetic, involuntary movements characteristic of tardive dyskinesia.
Scenario 3. You transition a patient to a highly effective atypical antipsychotic like clozapine to manage their treatment-resistant psychosis. The psychosis improves dramatically, but over the next 3 months, the patient experiences massive, unexpected weight gain and early signs of metabolic syndrome. Antagonism of which specific receptor subtype is most strongly driving this intense appetite increase? Take a second to think.
The answer is the 5-HT2C receptor. While 5-HT2A antagonism defines the atypical class and helps alleviate the psychosis, it is the potent blockade of the 5-HT2C receptor that removes the critical inhibitory brake on hypothalamic appetite control, driving the severe hyperphagia and subsequent metabolic disruption.
Scenario 4. You are reviewing the pharmacology of treatments for Alzheimer's dementia. You note that unlike dopamine, noradrenaline, or serotonin, the neurotransmitter acetylcholine does not utilize a presynaptic reuptake transporter pump to clean up the synapse. How is the synaptic action of acetylcholine primarily terminated? Take a second to think.
The answer is rapid enzymatic degradation by acetylcholinesterase. Acetylcholine lacks a vacuum cleaner. Instead, it is instantly cleaved into inactive choline and acetate in the synaptic cleft by ACE. This unique vulnerability is exactly why ACE inhibitors like donepezil are our primary pharmacological tool to try and prolong cholinergic signaling in Alzheimer's patients.
Final scenario number 5. A patient undergoing aggressive chemotherapy experiences devastating, relentless nausea. To provide immediate relief, they are prescribed ondansetron. What is the unique structural characteristic of the specific receptor this drug targets, which allows it to work so rapidly compared to other receptors in its family? Take a second to think.
The answer is that the 5-HT3 receptor is a fast, ligand-gated ion channel. Almost all other biogenic amine receptors, including all the other 13 serotonin receptors, are much slower G-protein coupled modulators. The 5-HT3 receptor, located densely in the emetic centers of the medulla, acts as a rapid excitatory electrical switch. Ondansetron physically blocks this ion channel, instantly shutting off the nausea signal without causing dopamine-related side effects.
Thank you for joining us on this incredibly deep dive. I highly encourage you to direct yourself back to Chapter 1.7 to review the detailed neuroanatomy charts and the receptor summary tables. We've built the conceptual framework and walked through the clinical reasoning. Now it's time for you to layer in the visual maps.
Happy study.
Here are the details of this episode.
Biogenic amine neurotransmitters. Byline: the modulators of mind-tuning mood, motivation, attention, and meaning.